And good morning, everyone. Everybody say hello. A small crowd. Anyway, so chapter 4 is basically the technological tools that we use, particularly looking at proteins. We're about to get into molecular genetics in terms of DNA and RNA and gene expression stuff. But a lot of work that we'll be talking about was done prior to the era of molecular genetics. And there's a lot of protein biochemistry that's sometimes now we view it as a bit quaint, but still useful. And there's a lot of terms and ideas that we constantly think about. And so one of the notions is this idea of protein denaturation. Chapter 4 is primarily about protein structure, which we've been covering in pretty good detail, I think. And as we were talking about the protein polymers, the idea that we have the primary structure, which is the amino acid sequence, and this is very easily predicted from the DNA sequence. And then from there, we can begin to interpret the secondary and tertiary structure, which is based on the order and composition of the amino acids, the R groups, and how they interact. So what this means then is that the secondary and tertiary structure is fairly delicate, the weak-ass bonds, and it's also critical for a protein's function. The way they fold up is how they work. So one of the things that we can begin thinking about is ways we can remove secondary and tertiary structure, and this is what we refer to as denaturation. So denatured proteins typically are not going to work. And we've been talking a lot about various kinds of systems and denaturants. A standard protein denaturant is, of course, SDS, the detergent. SDS, as a denaturant, is coating everything with a net negative charge, and so we end up having a protein that no longer has the charge interactions, and it will essentially unfold. Another part of denaturation that's important is that if you've got a portion of a protein that is a transmembrane stretch, this is a portion of the protein that is actually going to be embedded in the membrane, and that is going to have a stretch of hydrophobic amino acid R groups. So the hydrophobic side chains are kind of sticking out in the membrane, in the fatty acid layer. And in order to solubilize those, you actually have to use some kind of detergent because it acts as an emulsifier. So anytime we use detergents in cells, what we're doing is extracting as much of the protein as we can and denaturing the protein, so they typically are not going to be functional. I mean, there are some proteins that their secondary and tertiary structure is tough enough that they may be able to operate under fairly harsh conditions, but most proteins will be inactivated. Another standard denaturation technique is heating it up. Boiling is going to be a great denaturation technique. And we have all kinds of examples of what happens when you denature proteins this way. Very often, now with SDS, this acts as a solubilizing agent, but boiling is very often going to cause proteins to fall out of solution. So we can end up with an insoluble protein that you spin it down in a microcentrifuge tube and you have an insoluble pellet. And sometimes you can resolubilize these things, sometimes they can't. An example we talked a little bit about is like the protein ovalbumin, which is the primary protein found in egg white. And if you've ever cooked an egg, even very moderate temperatures cause the ovalbumin to fall out of solution. It turns white. And it's one of these proteins, actually, that is pretty much impossible to resolubilize after you've solubilized it, or after you've denatured it. So you can't get an egg white to go back into solution, try as you might. Other examples of protein denaturation that result in the protein losing its structure and falling out of solution are things like making cottage cheese. If you've ever done this before, it's actually a really delightful experiment in which you take milk, and the primary protein in milk is casein, and casein is quite soluble, but when you heat it up and then drop the pH down, ka-whoomp, it falls out of solution. And you end up with a soluble fraction, stuff that's still in the liquid, which is referred to as whey. And then you have the casein protein falling out of solution, which you then use a material known as cheesecloth to squeeze it out, and you can end up with a lump of cottage cheese. And when you do this, one of the things that becomes pretty obvious is that the amount of milk it takes to make a carton of cottage cheese is just insane, because it's multiple gallons to generate that little tub. But you can make little cubes of cottage cheese this way. So protein denaturation by boiling, by pH treatment, by detergent, all these things are rendering a protein basically non-functional most of the time. There are a couple of exceptions. A standard protein that we look at for protein structure analysis is an enzyme that is somewhat generically known as RNase. And RNase is a protein that will, as the name implies, degrade RNA. And it is, in fact, the number one problem when you're trying to isolate RNA is that RNases are everywhere, A, and B, they're phenomenally tough. Many RNases will be inactivated by boiling, but if you cool them back down, they fold back up and they go back to work. Now, most proteins don't do that, but some will. So RNase is a very tough protein, and you can inactivate it by boiling, but as soon as you bring the temperature back down, it's back to the races, begins chewing up your RNA. It's one of the things when we're talking about doing COVID-19 testing, because it's an RNA virus and you're detecting the RNA, the purification technique is a little bit tedious. In order to have the RNA present, you have to inactivate RNases, and because they are everywhere and so common and so tough, it's not a real easy thing to do. All right, so we've got a variety of different kinds of protein systems and different ways of manipulating them. Just as another thought, just to remind us, this idea of an antibody. This is a protein structure that actually has the ability to bind molecules. We'll be employing antibodies, at least in our lecture discussion. We don't use antibodies so much in the lab, just because. But antibodies bind to what is known as an antigen, that is, whatever you inject into the bunny rabbit to make the antibody. And it works outside of the organism, which, at least when I first started thinking about these, it was like, really? Yeah. You can actually take blood, spin the red blood cells out, have serum, and there'll be antibodies floating around in the serum, because they're actually fairly soluble, and it will still bind the protein that it was made against. And so this becomes an incredibly useful tool for identifying proteins. And you can use antibodies actually for identifying all kinds of things. So, various kinds of everything from a strep throat test to a pregnancy test to all sorts of strip test things are based on antibodies. So we use antibodies as a way of detecting proteins, and we were talking last time about protein gel blots, where you can run a gel out, blot it, and then use an antibody to detect the protein. But you can also use antibodies to detect proteins in the cell. And there you can look at at the protein localization within the cell. And that becomes a really important tool for understanding where some of these different proteins end up being on which side of a membrane or which side of a cell, things like that. Now, another interesting phenomenon in the protein biz that is related to denaturation has to do with proteins that can change shape and change their function. And it's not too many proteins that do this, but the number is increasing in terms of pathology and things. One example is a protein that is known as a prion. The prion protein. And has anybody ever heard of a prion? And do you know what it's related to? This is the causative agent of what is known as mad cow disease. Mad cow disease is a neurodegenerative disease that affects cows. As people began looking at this, there are a variety of diseases in different animals that have the same kind of pathology. And initially, they were viewed as separate diseases. In sheep, it's known as scrapie, but apparently the sheep start rubbing up against the fence posts and things and trying to itch themselves, and it's not very good for their wool coats. So scrapie. In deer, good marker, there is a disease known as chronic wasting disease. In humans, there is a disease known as Creutzfeldt-Jakob disease, known as CJD, and this is a lethal neurodegenerative disease. And it takes, quite a while, because these different kinds of diseases have been known for centuries. One final point, in Polynesian Islands and actually in Papua New Guinea, there was a neurodegenerative disease among the native inhabitants that they refer to as kuru. And it takes a while, up until like the 70s, before people began putting these things together and realizing that these are all the same kind of disease. And so there's a whole history of epidemiology and disease analysis from even back in the 19th century, a set of sort of rules known as Koch's postulates, which is just an interesting notion. It is a logic set, a way of trying to identify a disease-causing agent. And it's really, Koch was doing this, actually, I think with plants initially, where you identify a pathology, a disease pathology, and then you look to see if it can be transmitted, what is the transmitting organism? And so with a bacterium, you end up purifying the bacteria to a single colony and seeing, can it infect another organism? And if it does, do you see the same kind of disease pathology? If you do, you say, then you purify it from that organism and reinfect again. And it's a way of determining what is the disease-causing agent. So it's just a set of logic tools that you use. I was just actually listening to the radio. There's a new television series on 1980s London music scene when HIV was becoming a real serious health issue. And back in the 80s, when the virus that causes AIDS was being looked for, there was a whole set of logic steps that had to be followed to identify the specific virus. And we now know the virus that causes AIDS is known as the human immunodeficiency virus. And there was a range of different kinds of discussions about, was this in fact the virus that did it or not? And so it's really just a way of identifying the disease-causing agent. So with the virus, we have the virus, we know that's what it is. You can try to begin making a vaccine. For COVID, we're pretty successful. For HIV, not so much. Regardless, with prions, people have been trying to purify some kind of disease-causing agent. Fairly quickly, you can eliminate a bacteria, because there just are no bacteria being found in the brains of these dead organisms, or these sick organisms. I suppose if you took their brain out, they're dead, but regardless. People then began saying, this must be some kind of virus. And in fact, as late as in the early mid-'80s, prions were referred to as slow viruses because the infection and time frame it took to see the pathology was sometimes years. And the primary way that you actually do the diagnosis is post-mortem, in that you see essentially Swiss cheese holes in the brain. And so it is known as encephalopathy. Spongiform encephalopathy is the general term for this. And basically sponge brain, as opposed to SpongeBob. And all of these diseases exhibited encephalopathy, this spongy brain. And up until even the early 90s, which you have to realize I was doing my PhD back then, so I was listening to people doing seminars and stuff. folks were trying to isolate the virus that caused spongiform encephalopathy. So you have bovine spongiform encephalopathy, there's human encephalopathy, all these different organisms. Apparently you can, actually, it can cross species, so it's like, there's some kind of disease-causing agent here. And one of the seminars I remember seeing was a researcher who was working with hamsters, and apparently you could take brain homogenate from sheep who had scrapie, inject that into a hamster, and you could have an infection going on. And the way they would assay and assess the infections with the hamsters, when you go into the animal room, they would all come to the cage and stick their nose out. And if you boop them on the nose, a non-infected hamster finds that playful and wants to engage with you. But a hamster that has been, now begins to show the signs of the encephalopathy, will back off to the back corner of his cage. So they have a very sophisticated assay for this disease. And they then began looking for, and this guy was talking about how, he had given out like the third PhD that was a negative, looking for any kind of DNA or an RNA, anything that they could identify that was a viral contagion, and absolutely finding nothing. And so you fast forward a few more years, and a guy that actually ends up winning the Nobel Prize by the name Krusner, not that we need to really worry about him, he identifies a protein that is involved, and what he is able to do is actually using like a yeast model. He takes the protein, puts it into yeast, and this protein is able to adapt a couple of different, very stable three-dimensional structures. What we find is a protein that we now call the prion protein. It has a couple of different structures that it's able to adapt, or adopt, I guess. We have the normal structure, which, just for our little cartoon purposes, we'll draw as like a little piece of chewing gum. And then there is another structure that this thing can fold up to that is, in fact, the abnormal and, as it turns out, the disease-causing structure that folds up like this, like a little marble. So now, we've got this information. Here's a little marble. Now, what it turns out is the way this is actually transmitted is that the structure of the abnormal form can influence the structure of the normal form. If this comes into contact with a normal protein, it will cause the normal protein to change its shape and become the abnormal protein. So it is, in fact, a transmissible protein-to-protein based disease. And it turns out then that the abnormal disease-causing form of this is, in fact, very stable. It doesn't go back. You can boil it. It is really hard to actually renature it or pull it back. And so this is an example of a three-dimensional structure that you've got a couple of different kinds of thermodynamically stable structures. There's the normal one, but then we also have a disease-causing one. And this was the very first time that a protein structure change has been associated with a pathology. So once this was discovered, fairly quickly, a whole range of proteins that exhibit this have been identified. And if we think about this, this is pretty darn weird. Rather than having some kind of external disease-causing agent, you've got a naturally occurring protein within the genome being expressed in the brain, and it can adopt a disease-causing shape. And that disease-causing shape actually can be transmitted from one organism to another. So this is a really weird kind of situation. Now, it turns out that it's not all that unusual, in that there are a couple of other diseases there's another neurodegenerative disease, and they all tend to be this way. Neurodegenerative, that is Huntington's disease. And Huntington's disease is a neurodegenerative disease that is inherited, and it exhibits actually something known as anticipation, which is a really weird phenomenon. It took a while to figure out. And that if you have a grandfather that begins showing symptoms of this neurodegenerative disease at, say, age 75, the son might show the disease at age 50, and the grandson might begin showing the disease at age 20. And it turns out that with Huntington's, it has a stretch that is known as a polyglutamine stretch of amino acids. This is a set of amino acids, and the single letter code for glutamine is Q. You have a stretch of glutamines in this protein. Now, it turns out that if you have something like less than 10 of glutamines, that's going to be lethal, and we never see that in the population. The standard kind of range ends up being somewhere between 10 to 18 of these glutamines, and that's what you see in the population. Now, as you start to get to be, say, 19 to 25 glutamines, this begins to show the disease. And you can sometimes see even up into the 30s, this is going to be showing even earlier onset. So it turns out that during meiosis, the development of the egg or the sperm, you can have an expansion or a contraction, what's known as unequal crossing over, occurring in this stretch. And so this is how anticipation has been explained, in that in a single generation, you can go from having, say, let's go ahead and, grandfather has 19, and then the grandson, or the son has 25, and then the grandson has 35, and so you end up expanding this polyglutamine tract and generating the disease pathology earlier and earlier and earlier. And this polyglutamine stretch actually folds up into a disease-causing non-functional protein. So Huntington's does this. Other examples of protein shape change that we know, the protein that is involved in Parkinson's disease doesn't exhibit anticipation the same way, but it does have a repeat sequence, and it can fold up into an abnormal shape. There's even actually evidence for Alzheimer's. There's a protein that is known creatively as the tau protein, and it becomes, due to structural changes, insoluble. And so you end up with these plaques in the brain. So people have been actually now identifying a whole range of protein shape changes that exhibit disease pathology. Now, Parkinson's, Alzheimer's, Huntington's are not transmissible. So it's not like the same thing with the prion, where you had the malformed protein bumping into the functional protein and changing its shape. In this case, the proteins are, because of the genetics, are unfolding in a way that make them so they start to fall apart. Now, another interesting kind of twist to this, we mentioned this before, but we'll throw it out again. There are a class of proteins that are known as chaperones chaperone proteins. And previously, this class of proteins had been known as first heat shock proteins, and then just a standard kind of a variety of stress-induced proteins, because you could induce chaperone transcription and translation, induce the protein or the gene to turn on by taking cells primarily, but you can also do whole organisms, and giving them a heat pulse. So with tissue culture cells, it's a pretty easy thing to see. With organisms that don't maintain their body temperature, like insects, you can do this, where you raise the temperature up to like 42 degrees, would be maybe 103, 104. And that will induce these genes. For a long time, nobody knew what they were doing. What these guys are doing is they are involved in protein folding, refolding, depending on how you want your perspective of this. They are able to maintain and reestablish protein structural shapes. So it's one of the things we start talking about on Monday, the idea of transcription and translation. You begin to produce a protein from an RNA. And our model here is the protein's three-dimensional structure is such that different amino acids on this end of the protein are going to interact with amino acids on this end of the protein. They fold up. What do we have happening during the initial translation when the back end hasn't formed yet, and you got this guy saying, I need somebody to fold up with. I'm going to fold up with you. No. And so chaperones are maintaining the protein shape. I mean, I think of it as sort of like petting it and saying, they're there. The other side of the protein is going to be produced in a few moments, and then you can link up with your buddies. Just chill out. And so it, in a sense, coats proteins up and maintains their shape. And these heat shock proteins, they work because, or they are induced with heat because increasing temperature is going to subtly change the shape as you begin to lose secondary structure and tertiary structure because you begin losing some of the hydrogen bonds. So when you run a fever, you are in fact beginning to denature proteins within your body, which is one of the reasons why a fever makes you feel so weird. And heat shock proteins are right there trying to refold and settle everybody back down, trying to make sure that proteins fold back up into their most perfect functional state. Lots of really cool things can happen, particularly in model organisms like flies and yeast, in which you can essentially remove heat shock proteins. And when you do this, one of the things in flies is all of a sudden you see a whole set of different kinds of mutations and phenotypes happen or become visible that otherwise wouldn't be visible because the heat shock proteins are keeping things folded up properly. So the other side of this, and I haven't seen too much of this in terms of it getting to market exactly, but drugs for various things like Alzheimer's, Parkinson's, if we can induce these heat shock chaperones, we can begin to refold some proteins and actually begin to reduce the effect of things like Parkinson's and Alzheimer's. So this is one of the pharmacological avenues to try and reduce some of these diseases, is that you can induce heat shock proteins to try to refold stuff. Another way you can induce heat shock proteins is through dehydration, and alcohol will do this. So stress proteins come up, or these heat shock proteins come up not just with heat, but also exposure to ethanol does it. So it's probably a good idea to start now with your morning coffee, have a shot of bourbon, every morning as a way of constantly maintaining a level of heat shock proteins for keeping everything folded up properly. And the older you get, probably, multiple times during the day. It was just a thought. I do it, and I'm fine. All right, no. All right, so that is a lot of what we think about with protein structure. And let's see here. I've got my figures, and I don't know if you've got the book, it's nice to have, but let's see what we got here. I'm going to just throw a couple of different pitches up here. This is from the 4th edition of the book. And I probably will post on Moodle these PowerPoints. And it's one of the things I kind of do. We've been going on the board. And I like the board because it gives me an opportunity, or us an opportunity to draw a little bit, in that, particularly with complex kinds of diagrams and things, the textbook produces these really slick, airbrushed, computer graphic images that you can look at, but when you simplify them and actually draw them out, you can get better information from them. So I really am keen on this. But it's always useful to look at some of the pictures in the book as a way of thinking about it in different ways. I want to get close one. I hate closing the blinds because I love the sunlight. But, so, just in terms of different kind of protein types, we got enzymes. We have structural proteins. These are going to be proteins that are providing the overall mechanical support to cells and tissues. Transport proteins, and we'll be getting into these different kinds of systems as we start going. These are things that are carrying stuff, either between cells or from the external environment into the cell. So things like hemoglobin, they do this kind of thing. Motor proteins, I guess we're supposed to see here a guy that's about to use a motor protein in muscle cells to start running. So myosin is a major protein involved in actually changing cell shape. Storage proteins, we have milk and eggs. There you go. These storage proteins are typically found in the liver or in embryos, in that they are being used as a source of protein that we can then break down and turn into other things. Just sort of an interesting thing to think about from a cell biology perspective. When you're thinking about an egg, and a chicken egg or a human egg, the amount of material that's brought in by the sperm is really very, very small. There's virtually no cytoplasm being introduced by the sperm. All the sperm does is bring in the other set of chromosomes, so you have a diploid nucleus. But the egg has all the cytoplasm, all the cytoplasmic organelles. The mitochondria are all maternally inherited. And so you have within an egg, particularly like a chicken, which they're laid externally, and it's like, okay, dude, here's everything. Go to town because I'm out of here, and mom can get up and walk away. You have to take all that protein, break it down, reassemble the amino acids into a little baby chick. Kind of a cool idea. Signal proteins, we'll be talking about these in exquisite detail. Lots of different hormones and growth factors are in fact proteins. Insulin is a good example. Receptor proteins, things that like in the nervous system, some dude's eyeball, there you go. Another really important class of proteins are what we refer to as gene regulation guys. These are proteins that can actually turn genes on and off. I used to think that everything that you can think of is going to be explained by transcription control, turning genes off and on, in terms of like differentiation and stuff. And that meant that the genes that produced regulatory proteins were the really critical ones to look at. And these DNA binding proteins that act as switches, that was the stuff. I have now completely changed my tune, and that cell signaling is really what drives all of interesting developmental biology. And it eventually will get to turning on and off gene regulatory proteins. But this is not as big a thing as I had initially thought. Still, it's a very important part of developmental biology and regulation of cells. Then we've got special purpose proteins. They're all special purpose. Anyway, different organisms can make some really odd, unique proteins. Things like antifreeze proteins of our various cold weather fishes. One of my favorite proteins is green fluorescent protein from jellyfish. Let's see. And monilin. There is also something called thaumatin, which is a protein found in various plants that actually binds to the receptors on your tongue and tastes very, very sweet. So lots of different proteins. And we already kind of went through this in terms of making the peptide bond. Now, for my taste, this ball and structure is not nearly as useful, but we have the amino terminus, the central carbon with its R group, and the carboxy terminus. And we can begin, here's the R group of alanine. The R group of glycine, remember, is just an H. That is the simplest one, so that's what I always have memorized. We carry out a dehydration reaction or a condensation reaction, depending on how you want to talk about it, in which a molecule's water is generated, and we make a carbon-nitrogen bond, which we call the peptide bond, but it's just a plain old covalent bond. So, being able to diagram not the R groups, but amino acids coupled together is a really important thing to do, partly because by getting this basic sort of information and orientation laid out, it helps when we start thinking about RNA and the way we go from an RNA molecule to a protein molecule. It also helps to think about these different things in terms of hooking stuff together. What's the free amino end? What's the carboxy end? It's just a useful way to begin organizing the orientation of these different molecules. So I strongly encourage you to take a piece of paper, draw an amino acid, draw a second amino acid, and then couple them together with a peptide bond. Do that. The R groups, memorize it for biochemistry. So, as we have said, we've got a way of essentially categorizing the R groups. And then this is the different kinds of amino acids. We have a 3-letter code. Just as a note, the 3-letter code fits really well with DNA sequence because the 3 nucleotides, the codon, match up when you are drawing it out as a line. You can also use a single-letter code in which you put it in the middle of the 3 letters. There's glutamine, the poly-Q repeats, and we can then begin thinking about the side chains in terms of them being negatively charged, acidic, positively charged, basic. So I mentioned this probably, on Wednesday, or maybe on Monday during our snow day. A term that, in a way of thinking about macromolecules, is that we can say something is acidic. That means it's got a lot of negative charge. If something is said to be basic, it means it's got a lot of positive charge. So most proteins tend to be acidic to one degree or another, and that is, they typically carry a net negative charge. Proteins that are basic tend to be a bit more, not so much rare, but less, they make up a smaller percentage of the proteins in the cell. These are proteins that carry a positive charge. So that's a general way we think about sort of coupling pH and charge, is acidic proteins are negatively charged, basic proteins are positively charged. So we then have a whole class of amino acids that, depending on the pH, change their charge. They are polar, but they don't carry a specific charge. And then there's a whole set of non-polar amino acids, and these are guys that to varying degrees are going to be soluble in the plasma membrane and lipid and things. And so you can take a stretch of amino acids and by computer or by eye, sort of take a 10 amino acid window and scan down and begin characterizing these different blocks of amino acids in terms of their charge and their hydrophobicity. And you can begin to develop what's known as a hydropathy plot. That is, the parts of the protein that tend to be really fat soluble and parts that are going to be really, really water soluble. And so membrane spanning proteins are real obvious because they'll have a whole stretch of charged proteins, and then they'll have this big blob of very much nonpolar amino acids, and then you'll have another stretch of charged amino acids. And so this is where the protein can fit within the plasma membrane. So it gets to be a little bit wonky, but it's actually a really good way of looking at proteins and thinking about their function by looking at their structure. So this is just a little cartoon diagram of these different weak-ass bonds that are forming the secondary and tertiary structure. Things like electrostatic attractions, where we've got different kinds of charges interacting here. Not really an ionic bond, but an electrostatic kind of interaction. Van der Waals attractions, I view this as sort of the catch-all for if you're not sure what the heck is holding something together, it's probably Van der Waals forces. Hydrogen bonding is another critical part of this structure, but all of these are fairly weak bonds. So we've got the unfolded protein, and then when it generates its tertiary structure, it folds up, and we got this way of external regions are interacting with water, and the internal regions are kind of interacting with themselves, having their own little fat party. And this is how we begin to build a secondary and tertiary structure. So the idea of denaturations, urea is a chaotropic salt, it's called. It is a nitrogen, 2 carbons of nitrogen. I can't draw off the top of my head. It's actually what we generate as our nitrogen waste in our kidneys. Urea is able to interact with proteins such in a way that it can very easily denature proteins. Oftentimes, you can remove the urea and proteins will refold. So it is a pretty potent denaturant, but you can sometimes actually remove it and allow proteins to renature. Oh, there's urea. Thank you. All right. Let's see. So here is the idea of the prion protein, and oh, rats. As I was drawing this, I'm thinking, which one did they draw it? They had the normal protein as being the ball, and the abnormal protein is the piece of chewing gum. I like it the other way around, but that's me. And the model then is that sort of autocatalytically, the normal protein, if it touches the abnormal protein, can change its shape, and this is how we can begin to transition the normal protein set into this amyloid fibril, this aggregate that is insoluble and causes the disease pathology. Drawing it the way they did, yeah, it makes a lot more sense to stack up, regardless. Yeah. One of the things with the amyloid protein this way, it is really tough when it forms this shape, and even chaperones and heat shock can't renature it. So, sorry, it is pretty much still untreatable. Let's see. Oh, so this is the idea of how chaperones are working. They are interacting with proteins to try and correctly fold them. So you have some sort of odd shape here, and then you have a little and symbol. That's obviously the way it must be folded. If it was a smiley face, I would be sure it's folded properly. Yeah. And then we've got, these actually could be the base structure for a variety of Muppets, but no. You can take the amino acid sequence and begin building these three-dimensional shapes. And there's all sorts of different kinds of shapes floating around. You got little insulin over here. Collagen, which is the main extracellular protein in your skin, forms these really long fibers. Various enzymes. Here's an enzyme that binds to DNA, and we will invoke this notion of protein structure such that DNA binding proteins actually can feel around and identify a specific sequence. So it's easy to anthropomorphize proteins as little motor proteins that walk along a structure, but that's how it works. All right, so then there's different kinds of models. Take that for what you will. So here's ways of diagramming these different kinds of secondary structures. Don't worry about that. But here's the alpha helix, and then the beta pleated sheet, the zigzag. Yeah. Okay, so this is what we've been covering. Helices wrap around. I have no idea what this is trying to show us. Like, go left, go right, go left. And what's this bottom guy doing? Oh, you flip things around. But this is better. Zigzags. All right. And then, as we mentioned in our protein structure discussion at the end, there is this notion of quaternary structure in which you have two different proteins that interact. And because you can't determine that from the genome, I refuse to talk about it. Sorry. That's all I'm going to say. No, we have two different proteins that can now interact with each other through the same kind of weak structures and things. What else is here? Oh, the idea of the active site. So you've got a little fold, and between two different proteases, they both share the same little kink. Even though the rest of the protein doesn't have the same structure, they both are involved in breaking down peptide bonds, and not surprisingly, they both have the same kind of little active site spot. And more squiggles. And how many of you ever eat nerd rope? This is what this reminds me of. But, and then we have a virus that can auto-assemble and cause havoc. Anything good here? Not particularly. We'll be talking about a lot of these different molecules as we get into these different sections of the cell. We mentioned a little bit about disulfide bonds, and here's an example of two proteins linked by a disulfide. That's another way that we can begin building these larger complexes. Fine, whatever. Oh, it's resting in there. So one of the things that people then begin to do is look to see how the substrate or the ligand, that which is bound, interacts with the active site. Lots of fairly wonky, nerdy biochemistry. And this is an antibody diagram. Lovely. All right, some common functional classes. Memorize these? No. We'll be throwing out a variety of things like polymerases. We are talking about kinases, phosphatases, just a general term. Good. And last but not least, we've got the blueberry chewing on a piece of Twizzler, I believe. Yeah. Enough. How much more are you going to say? This is actually part of one of those back panels about how cells produce antibodies and antibodies recognize things. Yeah, and injecting a portable mouse, I guess. And let's see, monoclonals, very cool. Oh, using antibodies and molecular tags. We aren't going to do this, but there's some diagrams so you can see this. The transition state, fine. Here we have the, what is this? Not Twix, it is the pretzel wrapped around the cheese filling. Combo. The combo model of enzyme substrate binding. Some big ass molecule, feedback inhibition. And lovely. Actually, one of the more critical diagrams of looking at protein kinases Remember, they're adding a phosphate. That phosphate addition is turning proteins on. Phosphatases are removing the phosphate, and you can also have kinases that are inactivating proteins as well. All right. Really? How much more do we have? We talked about it. This is probably like one of my all-time favorite diagrams from the book. Is the protein is walking down the street. And then there's like the arm that's able to break into a safe. All right, so memorize these historical landmarks. And lastly, there's a 2D gel. We talked about that a little bit, but and big fermenters building something. How many more diagrams do we have? X-ray diffraction, I don't care about. Breaking cells and tissues. Anyway, this is all kinds of fun stuff that you can go back and look at. Oh, we'll talk chromatography. We really should be done here. Am I still recording? Sorry. Gel electrophoresis. We went through this. So these are some things. Oh, isoelectric focusing. Very cool. And a 2D gel. I love 2D gels. Another protein that's walking down the street with a trunk. That's the end of it. Hey, on Monday, we're going to start getting into the basics of genetics and turning DNA into RNA and RNA into protein. And we'll have a little homeworky assignment. And I've taught for 51